For this video clip, I have a special guest for you. First, Orlando Scherer from UNIST. Thanks for coming.
Nice to see you.
We have found the secret of life. That's what Dr. Francis Crick announced in a traditional English pub at the University of Cambridge in the United Kingdom on February 28, 1953. That morning, he and Dr. James Watson had decoded the molecular structure of DNA and solved the mystery of DNA base pairing. They concluded that G and C are linked together by 3 hydrogen bonds, like that, and A and T are linked together by 2 hydrogen bonds.
They were able to deduce the structure of DNA based on crystallographic data from Rosalind Franklin and Maurice Wilkins, which showed that the phosphate backbones were on the outside of the DNA and the base pairs needed to be on the inside of the DNA, and DNA forms a helical structure. Linus Pauling, who earlier discovered the structure of the alpha helix, a key structural element of proteins, had proposed a structure with the phosphate on the inside of the helix, but Watson and Crick were able to discount that based on the new crystal structure data. From the work of Erwin Schagraff, they knew that A paired with T and C paired with G, and using models that they moved around, they were able to find out how the bases pair with each other. They found out the binding pattern of the 2 base pairs and were able to propose a structure of the DNA double helix that was later proven to be entirely accurate by atomic resolution structure studies. As I noted previously, genes are equivalent to information databases, and the mission of DNA is to transmit that information. Watson and Crick also realized that their structure of DNA immediately suggested how DNA would be duplicated, namely with each of the strands serving as a template for a new strand leading to the generation of an exact copy of the original duplex DNA. Later in Unit 2, we'll see how that happens. The templating function of the duplex is also critical for the repair of DNA when bases are damaged by endogenous or exogenous sources.
So now, let's take a look at the structure of DNA backbone. You see the phosphate group and sugar. Substituents are tetrahedrally arranged about P atom right here. Then if you look at the oxygen here, these 2 oxygen atoms each share half negative charge, then it's quite acidic. Then here is a base right here. The 4 different bases are connected with this sugar phosphate phosphodiacetyl bond and the 2' dihydroxyribose sugar right here. Then you can see the base pairing right here, like this. Then AT base pair right here, 2 hydrogen bonds here. GC base pair has 3 hydrogen bonds right here, and they are all connected with sugar phosphate backbone. Then this structure is shared by all life forms. If you look at the structure like this.
What a beautiful structure it is. Look at this helix.
The 2 strands are intertwisted with each other like vanilla and chocolate in a soft serve ice cream cone.
This double helical structure helped us solve many mysteries of genetic inheritance that could not be explained by Mendelian genetics. As Dr. Watson explained, DNA is the substance that carries genetic information.
Therefore, the structure must be heterogeneous. On the other hand, the DNA can be crystallized. Therefore, DNA needs to have a certain uniform structure. So it is uniform and it is also heterogeneous inside. In the DNA double helix, its heterogeneous parts, actually base pairs, are facing inward, and its outside is made of a uniform, the backbone structure. There are a couple of different videos for the discovery of DNA structures. Please have a look at the different videos.
And in the next video club, we'll learn why DNA forms a helical structure.